Many of you have heard about ASM Strategic Roadmap, and if you have not, you got a good taste of it through the video that we just saw. I think it really encapsulates the structure that we're trying to set up at ASM in a new way, and how this positions us to solve the biggest challenges ahead of us. So we are very pleased today to present to you a unique and impressive scientific program that spans the microbial sciences. This is a field that is so diverse and yet so interconnected. At ASM Microbe 2024, we have nearly 400 scientific sessions for you to explore. So no matter what field of research is your primary focus here at ASM Microbe, I encourage you, each of you, to take full advantage of the wide range of sessions available and to explore something beyond your usual interests. So in the coming days, I invite you to attend sessions outside your area of expertise, ask questions, and engage in conversations with new colleagues in various disciplines. So we as ASM are a global community, and we have here today, or not here today, but at Microbe during this week, we have about 5,000 microbiologists from 95 different countries. And that's really just quite amazing about what we're all doing. So from our advocacy efforts to raising awareness about climate change, antimicrobial resistance, the bioeconomy, to making ASM and the microbial sciences more diverse, equitable, and inclusive, each one of you has committed to or contributed to the advances that we make in our field and also within ASM. So this year we have dedicated several sessions to exploring how microbes can be positively transform our world and the impact of scientific discoveries on our community and on our planet. So please don't miss the President's Forum on Saturday, where we will delve into the power of microbial sciences to change the world. Our second annual Science and Society keynote will take place on Sunday, and this will feature renowned infectious disease expert Peter Hotez in a conversation with MSNBC's Jonathan Kaper. So Hotez and Kaper will examine the importance of engaging with public on scientific topics and building trust in science. And I think we can all acknowledge that that's something we really need to focus on. So from sessions on drug discovery and the microbiome, to One Health and health disparities, to discoveries of new fundamental knowledge that make microbes click, this year's ASM Microbe is diving deeper than ever before into our constantly evolving and expanding field. As all of this would not be possible without the close partnership and collaboration of ASM members and the professional staff we have at ASM, who are all amazing. To this end, I would like to thank ASM CEO Dr. Stefano Bertuzzi and his invaluable staff and the ASM Microbe Program Committee for their hard work to make this meeting a reality. So I'd like to ask the Program Committee members that are here to please stand up to be recognized. And a very special thanks to our Program Committee co-chairs, Carla Satchel and Robert Tibbetts. We greatly appreciate your leadership. Would you guys stand up? Yeah. And to the ASM Meetings Department and staff that have worked so tirelessly to make all of this happen, and to their families that supported them through all of that, we thank you very much. And none of this would be possible without you. So, in addition, I would like to thank the ASM Board of Directors and ASM Council on Microbial Sciences for their ongoing leadership at ASM. And to our industry partners, we deeply appreciate your ongoing support. The exhibitors are an integral part of ASM Microbe who contribute to the excitement of our exhibit hall. And we want to give special thanks to our platinum sponsor, Diasorin, and we want to make sure that you will visit them on the exhibit floor at booth number 925. So with that, I would like to welcome to the stage ASM CEO, Dr. Stefano Bertuzzi.
Good afternoon, everyone, and so excited to see you here today. Thank you for being in Atlanta. Thank you for being at ASM Microbe, and really I also would like to give a big thank to the chairs of this meeting, Dr. Carlos Hatchell and Dr. Bob Tibbetts, who have prepared, as Virginia was just talking about, just an amazing program for you. Take it all in, enjoy. This is really the time where we all grow and we exchange ideas and thought for the future. What I will try to do in the next 10 minutes is try to show my enthusiasm and the enthusiasm of all the board of directors and the leadership of ASM for the great things that we think are happening at ASM. And we hope that this could be an invitation to all of you to be partners with us and work in making ASM as strong as it can possibly be. This journey that I want to mention to you today started almost 3 years ago, when we embarked in a project called Microbe 2040. And what we tried to do was something a little bit funky. And we said, let's imagine what microbiology would look like in 2040. And let's imagine what the world, what society at large would need from the field of the microbial sciences and from the microbiologists who are in this room. And of course, even the super smart people who are on the board of directors of ASM, sitting right here in front, they're my bosses, I need to say that, right? And, but it's true. They don't have a crystal ball. They don't know what the world will be in 2040. And that was an uncomfortable space to be. We are scientists, and we want to speak with the data. So where's the data? We don't know. So we took the approach of developing 3 different scenarios. An optimistic view, so what would happen if it's bliss, if everything that could possibly be right from here to 2040 actually would go right. A pessimistic scenario, what if everything goes wrong? And of course, the middle of the road. What happened after quite a bit of work on this, and we looked at this composite picture, and we thought, dear God, we don't know whether things will happen exactly in the way that we imagine in this 3 nicely packaged scenario. But what was very evident was that everything that was in here could happen. Everything seems really something poised to happen one way or the other. And so we thought, well, if this is really the case, and some of these scenarios are truly terrifying, and some of them are absolutely exhilarating, unleashing the power of the microbes to serve humanity, then shouldn't the role of ASM from here to 2040, that of actually helping steer the field toward the most desirable scenarios, the most desirable futures, and fend off those biggest threats that could be so scary? And that really resonated with all the people involved in that. The problem that became very apparent was that though we are not structured to do that, and like most professional scientific societies, we've been structured over the years to essentially think of ourselves as service providers. You need to publish an article. We have 7 fabulous journals where you can do that. You need to attend meetings. We have wonderful meetings like this to do that, and so on and so forth. And this is great, and this will continue. But in a sense, it is tactical. If you need to solve big problems like climate change, of any microbial resistance, of our issue, to really help steer that future, we are not structured to hold the space for doing that. And that is the roadmap that we deployed to try to correct this issue and embark in a different journey that Dr. Miller, our president, just mentioned. And so this is why we're launching at this meeting a new structure where we are building 3 different scientific units. ASM Mechanism of Discovery, building and harnessing the power of the fundamental microbiology, the fundamental science, ASM Health, and ASM Applied and Environmental Microbiology. This is really important, and as you can see here in some more detail, all the various areas that will encompass the various, the 3 scientific units. So why are we doing this? Why do we need this new roadmap? Well, number one is because we need to have a structure that is coherent, that is integrated and rooted in the science. And as you can see from the graphic, these are not Venn diagrams, these are not columns, silos. This is a circular argument. This is not circular argument, sorry. This is a circular graphic because actually it is about, to integrating and bringing them together within the whole microbiology field. Very important, these three units are important because they need to be the instruments. And pay attention, if you remember just one thing, remember this one. This is important because it needs to be the instrument that really helps empower you, the ASM members, the loving and caring microbiologists, to really help shape what the field needs to be. So we want, as a society, to put you in the driver's seat and have a platform through ASM that empowers you to say what you see needs to happen in your area of microbiology, not having others tell you what needs to happen, and then we as a society having to come and play whack-a-mole and correct what our members think was wrong. One way of seeing this would be moving from reactive to being proactive and having a structure that helps and empowers you. If we draw an example that always resonates with me in this from the field of economics, it would be moving from being price takers to being price setters. You decide what needs to happen. It's ambitious, I know, but this is what we're striving for, and this is why we're doing it, because of what I was saying before. We want to empower you to shape what the future needs to be and help steer toward the most desirable aspects possible. This construct would also allow us a very strong branding. One of the strengths of microbiology is its breadth. One of the problems of microbiology is its breadth. And very often we say, I don't see myself in this, right? And the three units we hope will help create that strong branding that is needed and strong independence in a sense of the community, but also integrating and interacting with all the other areas that are present at in ASM. A core principle of what we are doing, and we'll talk about this in a second, is all rooted in the core value that we embrace at ASM of open access, open data, and open science in a sustainable and equitable manner. And this is really key because we really want to make sure that we accelerate science as the maximum that we can, and as a very responsible publisher, one of the largest publishers of microbiology in the world, we want to be the leaders that really help move the whole field forward in this direction. One aspect that this strategic roadmap wants to leverage is the fact of ASM being the most inclusive and welcoming organization that is rooted in the geography of the country, of the United States, leveraging as much as possible our 35 branches, which are distributed throughout the states in the United States. And that is a terrific opportunity for creating that inclusiveness and bringing in all the people who may not be able often to attend national events like this one here on others. So that's a big strength that we're going to be leveraging in the next, in the future during our strategic roadmap. And the converse of that, which we need to keep both of them together, is the global aspect. And that is true because ASM is a global organization already. We have actually more members outside of the United States than in the United States. And it's also, we have a great network of ASM ambassadors that are doing great work. So really thank you, ASM ambassadors, for all that you're doing around the world. And we have a lot of activities that are going on in many, many areas of the world. So let me spend 1 minute or two about how the world tomorrow will look, and that why this global aspect of ASM that needs to be harnessed, it's so relevant for the future of science and microbiology in the world. If we look at 1989 at the United Nations data for population, and I looked at 1999 because that's the date of the fall of the Berlin Wall, which to me opened a whole new era in post-World War II. I don't have time to talk about it, But it is a really, really important time point, in my view, for understanding our past and our future. We had about 5 billion people on Earth. If we look at the projection from the United Nations in 2 years, we will cross the 8 billion people on the planet. And if we look 50 years from now, we will have over 10 billion people on Earth. This is a staggering number that poses tremendous challenges from a sustainability perspective. And what is important, even more than that, if you look in the third column here, is the distribution of the population over the world. In 50 years, we will have a doubling of the population in Africa. We will have a shrinking of Europe, and you see the data there. This poses enormous challenging from a microbiology perspective, from a sustainability perspective, political instability in general. Recently, a study from Goldman Sachs have looked at how the economies in the world will develop over the next 50 years. And if you focus on the column to the right, again, about 50 years from now, 2075, you see this list of the largest economies projected to be in 2075. When I first saw that, I jumped out of my chair, because look at the list. China, India, States, and then we have Indonesia, Nigeria, Pakistan, Egypt, Brazil. The first European country to enter this list is number 9 with Germany. In other words, in 50 years, we're projected to have a world that will look extremely different than what we have today. And so ASM is incredibly well poised in this new construct, which will be very complex and difficult for everyone, because this country that you see listed here are the countries where we have the largest number of members. And so we need to engage and embrace this and build an organization that really works around the globe in a way to help and microbial sciences in all these countries in order to really make an impact and solve those problems that we were mentioning. Looking domestically, I wonder, and this is my own personal considerations, where the current construct that we have for science in the United States is well prepared for this new world that I just quickly described. Our system is rooted in the wonderful vision that Vannevar Bush, who was the first scientific advisor to a president of the United States, Franklin Delano Roosevelt, who published this wonderful letter as a response to a letter from the president that was Science, the Endless Frontier, which actually was the one who established the National Science Foundation, and our Lifetime Achiever soon was the directors of the National Science Foundation. And in summarizing what the content of, from my perspective, the key elements of Science, the Endless Frontier of Vannevar Bush that built the scientific structure that we operate from in the United States, I see like 4 different tenets in there. The first one is, and I'm raising it, is that Vannevar Bush said all the science, something's happening with my microphone, all the science that was deployed during World War II that helped the United States win the war should now be used to win the peace. And this is what was actually happening. And this is actually what we're, the system that we're working in right now. And I think that this is great, but in moving forward, we need to harness and make it broader. And this concept of open access, open data, and open science need to be put on steroids. And also, it's very important that we have an integration and a collaboration among disciplines and among professional society to all contribute to shaping this new face of science. Also, a second element of that construct is this construct that the government funding the university for research, which is the core of our system, is fantastic, and it's what allowed us to do all the progress that we have made. However, in going forward, I think the relationship for science cannot be just between government and the university and the scientists. It becomes essential that the way we think about science engages all society, that science is part of societies, and that the the scientists really interact with society in science, making it part of it and an integral part of that. What is very important is the investment in the workforce, and that is absolutely important, and I would add in going forward, harnessing the diversity of the workforce in science is absolutely fundamental, and having the face of the scientist look like the face of society will be absolutely key. And the last point is that we need to look forward is really looking at the global aspect that I mentioned before, moving from a national system to really a global system that is integrated with the rest of the world. So these are my thoughts for what we have looking ahead. And I wanted also to now invite on stage our executive publisher, Melissa Jr., who will tell you how this concept and this work actually is going to be applied and worked on in our journals program, which is so fundamental. This is just an example of what we're doing and for the future, exemplifying this new construct that we're going to work around. So thank you very much for your attention, and Melissa, the floor is yours.
It's an honor to speak to you today, especially during these dynamic times in the microbial sciences and in scholarly publishing. I am thrilled to take a very brief 5 minutes to share two of ASM's latest initiatives that support the strategic vision that Stefano just laid out, and which highlight our commitment to equity, open access, and global scientific collaboration. ASM publishes 6 gold open access titles, freely accessible for anyone to read, and 6 subscription-based titles where content is gated unless an institution pays a subscription or if an author pays an article processing charge, otherwise known as an APC, to make their article open access. In 2025, we are launching our Subscribe to Open business model for these 6 subscription-based journals that will, in effect, allow for open access to all of ASM's research titles. The model has 2 primary components. The first is that S2O will allow all authors from any of our global subscribing institutions to publish in these journals without incurring APC charges. And then secondly, if our subscription revenue targets are met annually, we will make all content in each journal for that volume year open access perpetually, facilitating universal access to crucial research. This transition to S2O will enhance accessibility, foster collaboration, and accelerate scientific advancement within microbial sciences, all concepts that are core to open science and to ASM's mission. This transition to S2O, so if your institution does not currently subscribe, please do put in a request to ensure you have access to these benefits as authors and readers. In the same spirit of equity and accessibility, we are expanding our global footprint by hosting our first scientific symposium in China in partnership and collaboration with Tsinghua University in Beijing in September. Anchored by several of our journals, this event on microbes and human health marks a significant step in our efforts to engage with the scientific community in China. Beginning in 2025, we aim to host 2 annual events there, with plans to expand to 2 annual events in India as well. We are acutely aware that microbes know no borders, and microbial disease will remain a global public health threat. But we also know the power of microbes to solve humanity's most pressing challenges. By organizing scientific symposia in key regions worldwide, we hope to create platforms across the world for scientists to connect, collaborate, and advance knowledge across various fields of microbiology. We hope that these gatherings will facilitate the exchange of ideas and promote scientific excellence on a global scale. So, thank you. Please come visit us at the ASM booth to learn more about both of these initiatives in ASM's publishing program. And finally, as ASM's executive publisher, you can't expect me to not make a plug for all of you to publish in our journals. The journals we have a home for all of your research. Your research is our mission, and we deeply value our relationship with our authors. We not only champion academic excellence and integrity, but also ensure your research gets the most reach and visibility. We are striving to make microbial sciences more inclusive and equitable, and are thrilled to be taking these steps in that direction. So thank you for your time today, and I hope to see you throughout the week. And please enjoy your microbe experience. Thank you. And now I'd like to bring back to the stage ASM President Dr. Virginia Miller.
Well, many thanks to Melissa for telling us about all the exciting things that journals are doing for us these days. And I also want to thank Stefano for giving you a nice overview of the strategic roadmap changes that we're thinking about doing these days. If you have more interest in that, I'd like to invite you to continue the conversation about the future of ASM on June 15th at 2 p.m. in the ASM studio, which will be down on the exhibit hall. And we'll be joined by incoming President Teresa Kohler. So stop by ASM booth 209 for a deeper dive into the latest developments of ASM strategic roadmap and the future of microbial sciences. So for almost 40 years, ASM has offered a wide range of awards to recognize microbiologists for outstanding contributions to the microbial sciences via the ASM Award and Prize program. Every year, the awards and prizes are given out to exceptional individuals who have come from all subdisciplines and career stages of microbiology. The ASM Awards and Prize program is managed by the American Academy of Microbiology, and these ASM awards highlight individuals who have made significant contributions to advance the field, their careers, and their institutions. And I have to say that one of the most exciting things I got to do as president this year was calling these wonderful people up and telling them that they got their award. So we have slides that are showing you all the names of all the awardees, and so we need to hang. Congratulations. Okay, so big celebration. One more hand for them. Woo. People have done amazing things, and it's really a pleasure to be able to celebrate you up here today. So in addition to the awardees that we just saw, we want to introduce the fellows for the Academy of Microbiology. So the American Academy of Microbiology is the honorific society leadership group within ASM. And the mission of the Academy is to recognize scientists for outstanding contributions to microbiology and provide microbiological expertise in the service of science and the public. It fulfills its mission by election to fellowship in the Academy by convening colloquia and oversight of ASM awards programs and prize program. So to be elected to the American Academy of Microbiology is a great honor for any microbiologist. Fellows of the Academy represent excellence in their scientific area and are held to a high standard of integrity. Each year, the Academy elects exceptional microbiologists to fellowship through a very highly selective peer review process that is based on the records of scientific achievement, original contributions, and the record of scientific integrity that together has advanced the microbiology. This year, the Academy received 156 strong nominations for the Academy Fellowship. After 2 rounds of review, the Academy governors elected 65 fellows that hail from 7 different countries. Though not all of the 2024 fellows could come to the celebration in person today, please join me in congratulating the exceptional achievements of the class of 2024 fellows. So congratulations again, and I think we'll be taking pictures later after the end of the session, so thank you very much, and enjoy your new fellowship. So thank you. And finally, it's my pleasure to invite Dr. Jim Kaper to come out, and he will be introducing ASM's Lifetime Achievement Awardee, for you.
Each year, one microbiologist is identified from a large pool of highly competitive nominees to receive this high honor. The selection committee evaluated each of the nominees for their achievements, their careers, and the impacts that these candidates have made to advance the field of microbiology. For their lifetime contribution to science and the high ethical principles and values that this person upholds, the recipients of this award are examples of excellence and praiseworthy role models. Not only are they successful in their own career, but these lifetime awardees have also trained many exceptional scientists and influenced many more to pursue science and make meaningful contributions to society. The 2024 Lifetime Achievement Awardee, Dr. Rita Colwell, was nominated by Dr. Joe Zhu and supported by Dr. Jim Teege, last year's Lifetime Achievement Awardee, and also by another Jim, namely me. In discussing this nomination, a common reaction I heard was, hasn't she received this award already? No matter the timing, this award is richly deserved recognition for an incredible career of achievement in science, service, leadership, and mentorship in the field of microbiology and beyond. Just to note some broad strokes of her career, after receiving her bachelor's and master's at Purdue University, Dr. Colwell went to the University of Washington to achieve her Ph.D. in marine microbiology under the mentorship of Dr. John Liston. She started her faculty career at Georgetown University, and in 1972, she was recruited to the University of Maryland College Park as a tenured professor in the Department of Microbiology. She is now a distinguished professor at the University of Maryland and also holds a faculty appointment at the Johns Hopkins Bloomberg School of Public Health. Among her various academic honors and accomplishments, she has received more than 60 honorary degrees from around the world. Well worth applause. After her initial research start in marine microbiology, Rita pioneered some of the first bioinformatics efforts in microbiology with her work in numerical taxonomy and bacterial systematics. Her work in microbial ecology ranged from microbial degradation of petroleum and other environmental pollutants to the ecology of microbial pathogens in drinking and bathing waters, particularly in the developing world. Her work on environmental reservoir of Vibrio cholerae is particularly noteworthy, and we'll talk more about that in a few minutes. Rita's impact goes well beyond her research, with an incredible record of service and leadership at the national and international level. She has served as president of ASM, the American Association for the Advancement of Science, the International Union of Microbiological Societies, and other organizations. Among her many contributions to ASM, she served as the chair of the governors of the American Academy of Microbiology. And she transformed what had been a fairly sleepy honorary society for ASM into a very active and productive organization, generating many meetings and publishing reports on important topics. Perhaps most notably, Dr. Colwell served as the director of the National Science Foundation from 1998 to 2004, being the first woman and the first microbiologist to serve in this hugely important leadership role. Dr. Colwell is also an entrepreneur, having founded the company Cosmos ID in 2008. Cosmos ID is a bioinformatics company that provides a variety of sequencing, metagenomic, and phylogenetic analyses for a variety of environments and ecosystems. Rita has trained some 74 master's and PhD students, more than 50 postdocs, and numerous visiting faculty, fellows, and students from all over the world. And these mentees have in turn trained many others, and so her mentoring legacy exceeds to the hundreds and probably thousands of trainees. I'm very fortunate to be one of her PhD students. She has authored more than 800 scientific papers and reports and published 19 books. Her most recent book, A Lab of One's Own, One Woman's Personal Journey Through Sexism in Science, was published in 2020. This book is an inspiring and fascinating work that I highly recommend. There's much more that I could say about her accomplishments, but that would just reduce the time for you to hear from her directly. So without further delay, it is my honor to present to you this year's Lifetime Achievement Awardee, Dr. Rita Colwell. Well, it's wonderful to be here with you to honor this incredible lifetime of achievements. Then,
thank you very much, Jim, for the very, very kind introduction, and to the audience. It's great to be back at ASM, where my home is located.
You've had an amazing career, starting with training 60 Ph.D. students, 14 masters, and over 50 postdocs, and numerous other questions. So to start with an easy question, who's your favorite student? Don't answer that. I'm just joking. But could you talk about the importance of mentoring, starting with your own mentor, John Liston, and how that's influenced you and your mentoring style?
At the University of Washington, John Liston was an extraordinary individual. He was a Scotsman who had just arrived at the University of Washington to start a new program in marine microbiology. There probably weren't more than 10 in the whole world at that time. This was way back in 1958, 1959. But he was fascinating in that he was a person who enjoyed life in all of its forms, and he challenged me as his first student in this new program to be able to reach out and to explore parts of microbiology that I wouldn't have done before. But he also encouraged literature. We always had to read another of the great books. At lunchtime, we had to discuss it. So it was more than just microbiology. It was really sort of learning about the world. And I think what I've tried to do with my students, as you well know, I am not a hands-on person, but I certainly have enjoyed being able to bring the rest of the world to the students who were in the lab.
Well, although you weren't hands-on, you provided incredible opportunities to me and so many other students in your laboratory. Been speaking to some of your former mentees about questions that I might ask you. One asked, what factors informed your decision to invite anyone to join your lab? Because there's no obvious one phenotype.
I think what intrigues me is an individual who is curious about the world around him or her, and who has an independent streak, and that is the ability to work independently, to think independently, and not to be dependent on proscribed instructions. And so this means a certain kind of person who really can work well as a team, but also as a leader in himself and herself. And I think those students have manifested in the 64 students who are now out in the world, and delighted to have them there.
Oh, it's an incredible legacy. Another huge part of your legacy concerns your research on cholera and Vibrio cholerae. Your contributions have included establishing the intra-epidemic environmental reservoir of this organism, the correlation of environmental parameters such as water temperature, chlorophyll concentrations with infection rates, and even the use of folded sari cloth in Bangladesh to filter water and reduce transmission. Could you tell us a bit about what fascinates you about this disease and this organism, particularly in the context of safe water and just the bigger implications?
I think what's most fascinating about Vibrio cholerae is that it's a bacterium that's an aquatic bacterium, and it really is not designed, if you will, to be a human pathogen. It's just that we are sort of intermittent. We step into its role, or rather its environment, and its role, I think, is associated with its host, the invertebrate, the copepod. And I think the function, and this is a hypothesis I'm still working on proving, its function is really to osmoregulate, to assist its host in osmoregulation. But when it attaches to the gut of the human, it attempts to osmoregulate, of course, making us very, very ill, obviously seriously ill, and we are now in the 7th pandemic. What's fascinating is that we have been going through collectively the COVID pandemic, but simultaneously, in many parts of the world, we were in the 7th pandemic of cholera. And so understanding its natural environment, I think, gives us a handle on pandemics in general. And because the bacterium really reacts to its environment, temperature, salinity, the kind of nutrients that come in from disturbances in the environment, like a hurricane that stirs up sediment and creates a nutrient-rich environment. This allows us to use satellite sensing to predict the outbreaks of cholera, and we're doing that now for Ethiopia and Yemen and Malawi. So understanding the global aspects of a disease allows us then to provide global treatment, global prevention, and I think that's what's exciting about the bacterium.
It's a fascinating disease. I agree. But I'm going to move on beyond research into your incredible service and particularly your legacy as director, your term as director of National Science Foundation. So in starting that, you had two strikes against you. You were a woman, first woman to be in that position, and nearly all the previous directors had been physicists or engineers. So what challenges did that present, and how did you deal with that?
Well, first of all, the National Science Foundation is an extraordinary organization. That was one of the best experiences of my life, to be the head of the National Science Foundation. But indeed, it was different to be a woman director, and especially the previous directors had all been physicists or engineers in the so-called hard sciences. Well, I think microbiology is a pretty hard science too. And what was excellent about being the director is I love science, all of it. And so I was able to assist in developing telescopes for astronomy and some of the apparatus for some of the breakthroughs in physics. But more importantly, some of the things that we were able to establish were biocomplexity. Now, that's something that's kind of interesting. Going to Congress and suggesting that we needed an investment in the environment wouldn't have been very, very well received. However, by explaining that, it's very complex to understand the natural environment, and so we really need to invest in biocomplexity. Well, that was great because I got almost $1 billion new for microbiology and the environment. But I think what was probably the most difficult part was that going to Congress to defend the budget, fortunately, I had a wonderful deputy director, Joe Bedonia. He was an engineer. He was the dean of engineering at Penn. And so the two of us would go to the Hill. And presenting the budget, the chairman of the Appropriations Committee would address questions to Joe. And Joe very nicely would say, well, I am sure the director would like to answer that question. So we got on very well, and fortunately, we were able to obtain really big increases, 14, 15% increases. I wish we could do that today. It's more difficult.
Well, it's quite a record, quite a legacy you left at the NSF. I want to talk about, turn to your book, your memoir. It's subtitled A Woman's Personal Journey Through Sexism in Science. It's an inspiration to all women scientists and to all men who support women scientists and women in other endeavors. But there's many possible questions I could ask you, but let me just start with a broad question that you could take anywhere you want, which is, what motivated you to write this book?
Right now, I think that we still have a lot to do to ensure that women are recognized for their talent, their contribution, and their role in science, engineering, medicine, mathematics, for example, as well. So I think there's still work to do. We've made changes. Things aren't the way they were, 40, 50 years ago when I requested to be considered for a fellowship and was told by the chairman of the department, we don't waste fellowships on women. You'll just get married and have children and disappear. Well, a lot of us here in the audience have shown that that's not exactly what we do.
When I was in your lab, it was the late 70s. You were on the ascending slope of your career. and you experienced many instances of sexism, but I don't recall you ever, quote, bringing that into the lab or complaining to us about that. The alternative hypothesis is that I was a clueless young man who wouldn't have recognized it anyway, but I don't think so. Remember, I brought to your attention the issue of this notorious ASM session decades ago where the convener really bullied and just treated you terribly then. But you handled that you were very forbearing then, and where other people would not be. What do you explain your approach in terms of doing that when faced with such displays? And
Well, I think it takes a good sense of humor, and I had to learn early on that it doesn't help to get mad. You just have to sort of get even.
That's been very helpful, obviously, throughout your career. Your approach has really, the evidence is obvious. But another time, I want to go back to the NSF director with the anthrax attacks in 2001. And you coordinated the response of many government agencies to the anthrax attacks. In your book, you cite many examples where women aren't at the table or are allowed to be leaders. But yet this situation was a notable exception to the rule. Can you tell us a little bit about that?
That was extraordinary. I'm sure very few in the audience knew that there was a committee which I was asked to chair because I had been doing a good deal of work with the intelligence agencies. So I chaired this, we called ourselves the National Genome Sciences Coordinating Committee. We could have had a presidential directive, but then we would have been FOIA'd, Freedom of Information Act. The FBI and the CIA would not have attended because they had a criminal case to solve. That is, to find out who was sending out these anthrax letters that was making people ill and killing people. We worked together. There were 17 agencies, and I chaired this. There was the NIH, the NSF, the FDA, the DOD, all of the agencies, and we met once a week, Friday afternoon. We had to meet between 1 and 2 because the CIA and the FBI lab directors were in Virginia, and the traffic would be very bad if we left after 2 o'clock. But we met every week, and for over a 3-year period, this was very early on now. This was before we had not very much sequence. We had perhaps 2 or 3 strains had been sequenced, and only half the sequence of Bacillus anthracis had been done. So what we did was pull together as quickly as we could with this team of people from all the agencies, the funding that allowed us to purchase the equipment. We bought instruments for CDC, for example, to sequence the smallpox, because what we did was build what is now the list of the threat agents, the most serious down to the ones that really could be economically treacherous. Hoof and mouth disease, for example, if that were released, that agent were released in the west of the US, it could have been disastrous. But over that period of time, working together, we were able to build through sequencing, we were able to track the source. We'll never know the perpetrator fully because the individual who was targeted, the day he was to be arrested, he committed suicide, sadly. But it was an extraordinary experience working together, all the agencies of the federal government, chairing this group, and being able to come up with a solution. That showed me, actually, that collaboration in science is very powerful. And I think that's a message we should really consider at the present time.
Yeah, thank you. I see we're almost out of time. So many other questions, but just to finish up, you've had an incredible career, and you're still going strong at an age when most of your contemporaries have long since retired. We've heard a bit about some of the hardships and the challenges that you've encountered. But what is it in your daily life as a scientist that brings you joy?
Well, I have to confess that my children give me the greatest joy. I have 2 daughters. One is a physician, the other practices medicine Canada. And the other is a botanist who is now an expert on wildflowers at the University of California, Davis. So it's wonderful to have had a fantastic family, wonderful husband, children, and wonderful students. So,
Thank you.
and now I have grandchildren.
There you go. Well, thank you, Rita, not only for this afternoon's stimulating discussion, but for the many highly significant contributions you made to microbiology and to science in general. Congratulations on this award.
Thank you, Jim.
As the Basic Sciences co-chair of the program committee, I'm honored to collaborate with an amazing group of dedicated colleagues who have worked to create an outstanding lineup this year. The program committee is a team of academics, scientists, physicians, and laboratory directors who volunteer their time and expertise to design, develop, and implement the scientific sessions you will hear at Microbe over the next 4 days. And I think we have a really exciting agenda ready to go for you, and I hope you enjoy the next 4 days. But this evening, I get the great pleasure of introducing the opening keynote speaker at this opening session. Dr. Emily Balskus is originally from Cincinnati, Ohio, where she became interested in chemistry as a high school student. She graduated from Williams College in 2002 as valedictorian with highest honors in chemistry. After spending a year at the University of Cambridge as a Churchill Scholar in the lab of Professor Stephen Ley, she pursued graduate studies in the Department of Chemistry and Chemical Biology at Harvard University, receiving her Ph.D. in 2008. Her graduate work with Professor Eric Jacobson focused on the development of asymmetric catalytic transformations and their application in the total synthesis of complex molecules. From 2008 to 2011, she was the NIH postdoctoral fellow at Harvard Medical School in the laboratory of Professor Christopher Walsh. Her research in the Walsh lab involved elucidating and characterizing biosynthetic pathways for the production of small molecule sunscreens by photosynthetic bacteria. She also received training in microbial ecology and environmental microbiology as a member of the Microbial Diversity Summer Course at the Marine Biology Lab at Woods Hole during the summer of 2009. Dr. Balskus joined the Chemistry and Chemical Biology Department as a faculty member in 2011 and is currently the Thomas Dudley Cabot Professor of Chemistry and a Howard Hughes Medical Institute investigator. She focuses on deciphering microbial mechanisms and its role in complex microbial ecosystems like the human gut microbiome. Using chemical knowledge and tools, Dr. Balskus and her group seek to discover new microbial enzymes and metabolites, as well as elucidate their biological roles. The research program has been recognized with multiple awards, including the 2011 Smith Family Award for Excellence in Biomedical Research, the 2012 NIH Director's New Innovator Award, the 2013 Packard Fellowship for Science and Engineering, and the 2019 Blavatnik Award for Young Scientists in Chemistry, and the 2020 Alan T. Waterman Award from the National Science Foundation. In this evening's presentation, Dr. Balskus will describe how microbes are providing exciting and unprecedented opportunities for the discovery of new enzymes and metabolites that have important biological function. She will identify emerging approaches for mining microbial genomes and microbiomes to uncover enzymes and molecules. As she will discuss this, as well as new methods to understand their biological impacts with a focus on the human microbiome. Please join me in welcoming Dr. Emily Balskus to the stage to present her featured scientific lecture, Chemical Discovery in the Microbial World.
So 14 years ago, I actually attended my first ASM meeting in San Diego towards the end of my postdoc. And this was actually my first time attending any type of conference outside of the field of chemistry. And it actually had a really big impact on me and the direction I've taken my independent scientific career. So I was not just excited and captivated by the science that I heard at that meeting, but I also felt very welcomed and encouraged by this scientific community, even with my very distinct scientific background. So, especially to the young scientists in the audience who may be attending ASM Microbe for the very first time, a special welcome to you. And I hope this meeting also has an incredible impact on your scientific journeys. And so, as was highlighted in the introduction, my training was originally in synthetic chemistry. And when you think about chemistry, I'm guessing most of you probably imagine something like this. Of course, this is what I thought chemistry was when I was at the start of my scientific career, training as a chemist, learning how to build molecules in a lab. But my scientific career obviously changed when I became exposed to microbiology and realized that some of the most talented and innovative chemists have never worn a lab coat. And of course, I'm talking about microbes. So in addition to being the most numerous organisms on the planet, living in almost every conceivable habitat, spanning a huge breadth of phylogenetic diversity, microbes are truly amazing chemists. And we are impacted by their chemical capabilities every day. And so it was the chemistry of microbes that first drew me into microbiology, specifically the rich opportunities they provide for the discovery of new enzymes and metabolites. But in part inspired and encouraged by that first ASM meeting and the many microbiologists that my lab and I have had the privilege to interact with since, we've also become fascinated by the biological roles that microbial metabolism plays in complex microbial communities, with an emphasis on the human microbiome. Microbial metabolism expands the chemistry that occurs in the human body and mediates important interactions with the host. However, our understanding of its biological consequences is still quite limited. Known metabolic activities associated with the microbiome typically haven't yet been linked to specific genes or enzymes. A huge fraction of both the genes and metabolites associated with the microbiome are still uncharacterized. And we're only just beginning to elucidate the molecular mechanisms by which microbial metabolism influences the health of the host and the rest of the microbiome. And so the goal of my lab's research is to use chemical knowledge and approaches to discover metabolic activities and enzymes from microbes, including the human microbiome, and to decipher how these activities influence host biology and other microbes. We hope that by gaining a molecular understanding of microbial metabolism, we will both reveal new fundamental biological insights about the microbiome, but also perhaps in the future enable therapeutic intervention. And so specifically, we think that by connecting microbial genes and encoded in genomes and metagenomes to the proteins they encode, elucidating the functions of those proteins, including enzymes that make metabolites, we can greatly improve our ability to interpret multi-omics data, specifically to infer functional information accurately. We think that this type of work also illuminates strategies we can use to try to manipulate functions in microbiomes. For example, by engineering microbes and introducing new functions into microbial communities that may lack them, or by developing inhibitors that target specific microbial enzymes and might be able to prevent specific types of transformations within these communities. So over the last 13 years, my lab has been studying a wide range of different microbial enzymes and metabolites that have expanded our fundamental understanding of enzymes, but also revealed some very intriguing links to human health. Now, many of these projects, perhaps unsurprisingly, focus on chemistry that involves dietary or host-derived molecules. For example, we've studied the metabolism of the notorious disease-associated molecule cholesterol and discovered enzymes involved in converting cholesterol to a poorly absorbed sterile caprostanol. So this activity, which is not performed by the host, is instead carried out by uncultured gut bacteria, may be beneficial for the host as it's correlated with lowered cholesterol levels. So if you're interested in learning more about our latest work in this area, a current postdoc in my lab, Antonio Tinoco Valencia, will be speaking on Monday about his efforts to characterize additional enzymes that are involved in caprostanol formation. We've also studied how the gut microbiome metabolizes small molecule drugs, including the important Parkinson's disease medication, levodopa. It turns out that gut microbial metabolism of levodopa reduces its bioavailability, likely restricting the amount of drug that can enter the brain where it's needed. And so this pathway not only has potential implications for better understanding patient response to levodopa and side effects, but also involves some unusual metalloenzyme chemistry. But in addition to metabolizing these ingested molecules, gut microbes can also make their own unique bioactive metabolites, often termed natural products. And it's this class of molecules that I'd like to focus on for the presentation today. So natural products are an important source of drugs, particularly antibiotics. They are also really important tools for research. So we may use natural products routinely in many of our laboratory experiments. And importantly, studying natural products and elucidating their targets has historically been a really important catalyst for learning about fundamental microbial and mammalian biology. And so my group and I have been very excited by the opportunity to begin to study natural products that are being produced by human-associated microbes. So in keeping with the spirit of this session, I'd like to tell you today about one project that really shows how investigating gut microbial natural products can illuminate new biology. I think this project, which actually started with an interest in a primarily chemical question, has also taken us in many unexpected scientific directions. And I hope that this adventure will capture your scientific curiosity and imagination. Okay, so this story began in 2006 with the discovery by Eric Oswald's lab in France that human gut bacteria, specifically Enterobacteriaceae, including many strains of E. coli, that harbor a group of uncharacterized genes called the PKS gene cluster, could induce DNA damage when co-cultured with human cells. This finding caught my attention because of the possibility that this type of gut bacterial activity might have the potential to contribute to colorectal cancer. Colorectal cancer is one of many intriguing links between the gut microbiome and disease. And over the last several decades, multiple types of microbes as well as microbial functions have been correlated with colorectal cancer, including the gut bacteria that carry the PKS genes. So for example, there have been studies that have been reported that gut bacteria carrying these genes, so-called PKS positive bacteria, are found much more frequently in colorectal cancer patients, and these genes have been shown to play a causal role in tumor progression in multiple mouse models of colorectal cancer. So this accumulating evidence linking these PKS genes to colorectal cancer really underscored the importance of figuring out what the functions of these genes were. And so if you analyze the genes encoded by the PKS gene cluster, they turn out to encode something called an enzymatic assembly line. So just like we use assembly lines to build cars and other complex machines, enzymatic assembly lines, like non-ribosomal peptide synthetases and polyketide synthases, use the coordinated action of multiple enzymatic domains to build complex molecules. And so it was very reasonable to propose upon the discovery of the PKS gene cluster that the enzymes it encoded were probably building some type of complex genotoxic natural product. So the presumed product of this gene cluster was given the name Colibactin. And so at this point, what a chemist would normally do would be to use Colibactin's genotoxic activity to guide the isolation and structural characterization of the natural product before trying to study its bioactivity or biosynthesis. But despite attempts by my lab and many other groups around the world, this conventional approach has completely failed for Colibactin. There has still been no successful isolation characterization of this metabolite. You'll see later that we think this is likely due to the chemical instability of this molecule. So my lab and I decided to take an unconventional approach to try to solve this problem. We wondered, what if we could study Colibactin biosynthesis before isolation? What if we could use bioinformatics, our understanding of other enzymatic assembly lines, and our chemical intuition to make predictions about what chemical transformations these enzymes should perform? We could then imagine testing these hypotheses by characterizing the activities of the purified enzymes. And so using this reverse approach, my group was actually successful in gaining the first information about Colibactin's structure. And this renewed interest in this natural product and inspired other labs around the world to join us in this effort. So after many years, the hard work and creativity of an amazing team, graduate students Carolyn and Lee and postdocs Matt and Yindi, provided enough information to allow us to propose a complete biosynthetic pathway and chemical structure for Colibactin. So on the next slide, I will show our structural prediction, which again, we formulated from studying biosynthetic enzymes in vitro, but also characterizing products that accumulated in mutants that were missing individual PKS genes. Now, also want to point out that this structure is consistent with that proposed by the Herzen and Crawford labs using complementary methods. Now, this is obviously a very complicated chemical structure, and I'll tell you a little bit more about how it is biosynthesized later on in the presentation. But for now, I just want to highlight a few important structural features that we discovered in this effort. First, you'll note that Colibactin is a pseudodimeric molecule, so it's almost symmetrical, but not quite. The other really intriguing structural feature of this molecule is the presence of these cyclopropane rings. Now, this was very unexpected because the PKS gene cluster did not encode any known cyclopropane biosynthetic enzymes. So it turned out that these rings are made using a completely new biosynthetic strategy that we were able to elucidate. So to share some of what I think is some really neat chemistry, what we discovered is that the enzymatic assembly line has actually evolved to use the cofactor S-adenosylmethionine as a building block for amide bond formation. So a specific enzyme, CLBH, loads SAM onto the assembly line, catalyzes amide bond formation, and then a PKS enzyme, which would normally do completely different carbon-carbon bond-forming chemistry, generates the cyclopropane by likely first forming a thioester enolate and then doing an intramolecular alkylation. Now, the reason I think this is interesting is because it's a new role for the cofactor SAM. We typically think of SAM as a donor of methyl groups, but it is also a non-proteinogenic amino acid, and so it makes sense that nature might have evolved the ability to use it for this type of assembly line chemistry. But of course, it wasn't just the chemistry of the cyclopropane biosynthesis that made discovery of these structural motifs so exciting to us. The presence of these strained, potentially reactive rings in Colibactin gave us a critical clue as to its bioactivity. Specifically, it suggested that Colibactin might be reacting directly with and damaging DNA. So this emerging understanding of Colibactin's structure allowed us to move forward and begin to address several of the important biological questions related to this natural product. One goal that we had was to start to decipher the mechanisms by which Colibactin might be affecting the host and potentially contributing to cancer. And this was an effort started by Matt, Yindi, and Paul, and most recently led by a postdoc, Eric. We and others had been recognizing, as we started to understand Colibactin's structure, this potential to react with DNA. So even before we had a complete structure prediction, mechanism for its contribution to cancer was beginning to emerge. We thought perhaps through DNA alkylation and then misrepair of the resulting DNA damage, Colibactin might perhaps be generating cancer-promoting mutations in the host genome. Now, to start exploring this hypothesis, we first sought to obtain direct evidence that Colibactin could indeed alkylate DNA in biologically relevant settings. Our first goal was to actually try to identify the products resulting from this DNA damage. So we sought to identify and characterize Colibactin DNA adducts. Now, this turned out to be quite challenging for a number of reasons. First, DNA adducts are typically formed at very low levels in cells, and so most methods to detect them using techniques like mass spectrometry are targeted. You have to know the structure of your adduct in advance. We didn't understand the complete structure of Colibactin, so we had to find methods that would be both sensitive but also unbiased. Secondly, and this is actually a challenge that applies to almost all of our work with Colibactin, because of the chemical instability of this natural product, we can't just buy it from a chemical supplier or isolate it and treat cells with purified Colibactin. We actually have to use the living microbes as a source of this metabolite. So that introduces significant experimental obstacles. Luckily for us, we were able to team up with a toxicologist at the University of Minnesota, Sylvia Balbo, who had been developing methods for detecting DNA adducts in untargeted ways, specifically the development of untargeted DNA adductomics methods. So with her help, we were able to compare the DNA adducts generated in human cell lines exposed to Colibactin-producing bacteria or isogenic mutants missing this pathway. And this comparison revealed two very promising initial candidates for these adducts. And so these two adducts, which have the same mass, we hypothesized they were likely isomers of one another. We were able to structurally characterize them using mass spec and NMR, and this did indeed reveal that they derive from ring opening of that proposed cyclopropane by a nitrogen atom of the nucleobase adenine. So this not only confirmed that Colibactin was alkylating DNA, it also gave us some important structural information about the natural product itself. Looking at this chemical structure, you can kind of see this roughly corresponds to half of that pseudodimeric molecule. Indeed, we were able to then show that these DNA adducts were coming from decomposition of a larger interstrand crosslink. We did this by characterizing larger adducts and also detecting interstrand crosslink formation in cells exposed to Colibactin. Interstrand cross-link formation is, of course, consistent with that proposed structure, which contains 2 reactive cyclopropanes. I think it's also worth emphasizing that interstrand cross-links are very potent and problematic forms of DNA damage. So finally, working together with my colleague at Harvard, Wendy Garrett, we discovered that these DNA adducts form in mice that have been colonized with Colibactin producing E. coli. So this really reassured us that the chemistry that we had been studying in lab in a culture flask was also relevant in a host setting. And it also highlights a biomarker that we and others can now start using to detect exposure to Colibactin in human samples and perhaps study its role in cancer development. And finally, most recently, in some unpublished studies, we've been elucidating the specificity of Colibactin DNA alkylation. So this has involved finding short oligonucleotide sequences that can undergo a reaction with Colibactin. And then we've used various analytical methods, including mass spec, to deduce exactly where the cross-linking has occurred. We can then readily vary the sequences of oligonucleotides to elucidate the specific features required for cross-link formation. And these efforts have revealed that Colibactin does not just indiscriminately cross-link DNA oligonucleotides. It actually has a strong preference for cross-linking 2 adenines that are 4 base pairs apart within AT-rich sequence contexts. So it is a highly specific DNA cross-linking agent. Now, this molecular information that we've been gaining about Colibactin-mediated DNA damage has been helping the efforts of others who've been interested in connecting this microbial genotoxin to specific mutations in the human genome. For example, in 2020, multiple labs identified mutational signatures, unique mutations, that are associated with Colibactin exposure. These mutations turn out to occur in a sequence context that we now know corresponds to the site of DNA alkylation that we've characterized experimentally. So this work, I think, is really quite exciting because if you think about it, these mutational signatures serve as a molecular record of human exposure to Colibactin. And indeed, these and subsequent studies have found these mutational signatures are indeed present in cancer genomes and found in genes that are known to be driver genes in colorectal cancer development. So collectively, this work really does demonstrate that humans are exposed to Colibactin and really highlights a need to further investigate the implications of this DNA damage and these mutations in the context of cancer development. And so, given these findings, we really sought a way to control Colibactin production in complex microbiomes in order to better understand and potentially prevent its contributions to disease. And to do this, we decided to develop small molecules that inhibit Colibactin biosynthesis. I think this is a great place to highlight just how challenging it is to manipulate individual microbial functions in complex microbiomes. So we think about applying tools like antibiotics. These will certainly change microbial metabolism and functions, but will do so in a broad way. The types of tools we'd like to develop, small molecules that inhibit specific microbial enzymes, we think would be a very complementary approach to microbiome manipulation, one that might allow us to elucidate roles of individual enzymes and metabolic activities within these complex habitats. Also potentially, I think, an intriguing therapeutic strategy to consider. And so the work on this project in the lab was led by former graduate student Matt. It has been a close collaboration with my Harvard colleague Rochelle Godet and her former graduate student Jose. And the question in beginning this project was really, what's the best target? How do we best interfere with Colibactin production? And this led us back to the very first enzyme we had actually studied in this pathway, an enzyme called CLBP. So CLBP is a serine peptidase. It's actually located in the periplasm of bacterial cells, so outside of the cell. And it catalyzes the final step in Colibactin biosynthesis. So it takes a precursor that we named pre-Colibactin and specifically hydrolyzes two N-acylated D-asparagine motifs highlighted in red from the ends of that inactive precursor. So this amide bond hydrolysis unmasks two primary amines, and this actually triggers a beautiful cascade of cyclizations that ends up dramatically altering the reactivity of the cyclopropanes, making them much more electrophilic and better at alkylating DNA. So we think this clever biosynthetic strategy, the idea of making an inactive precursor, or a prodrug, exporting it out of the cell and constructing the reactive warheads in the periplasm, probably evolved as a way for the producing organism to protect itself from Colibactin. So we wondered whether it might be possible to prevent Colibactin biosynthesis by inhibiting the activity of CLBP. So to do this, we knew about the reactivity of this enzyme and rationally designed highly selective small molecule inhibitors that actually mimic that substrate pre-Colibactin, specifically that N-acylated D-asparagine structural motif. We appended a boronic acid onto these inhibitors. This group is electrophilic, and we hypothesized it would engage the catalytic serine of CLBP in reversible covalent bond formation, disrupting enzyme activity. And these rationally designed inhibitors worked really well, so they are quite potent both against the purified enzyme and also in bacterial culture. And together with Rochelle, we solved a crystal structure of CLBP in complex with these inhibitors. This showed that indeed they are making a covalent bond to that serine. This structure also highlights some interesting features of CLBP's architecture. You can see that it's actually anchored in the inner membrane. And you can see the active site actually spans these two domains, the catalytic domain and the transmembrane domain. And we think that the acyl tail of that N-acyl asparagine motif probably extends down into the transmembrane domain. This structure also revealed something that was quite intriguing, the fact that CLBP appeared to be dimeric. So you can see here the dimer interface that I've highlighted here. It's quite extensive. To really demonstrate that this was not just an artifact of crystallography, Jose also solved a cryo-EM structure of CLBP that again showed this dimeric architecture. Now, the reason why this is so interesting has to do with our proposed structure of pre-Colibactin, the substrate for this enzyme. Remember, we think pre-Colibactin and Colibactin are pseudodimeric molecules. A CLBP dimer would have two active sites and two catalytic serines, so might be capable of processing both warheads at once. So indeed, our proposed structure of pre-Colibactin into this dimer, we see that it fits perfectly, suggesting that perhaps the formation of these two cyclopropane warheads is coordinated by the enzyme. And I think this lends further support to our structural predictions. Now, in addition to looking at the inhibitor in bacterial cultures, we also wanted to explore its utility in complex settings. So we did this, the inhibitor, with a complex microbial community ex vivo. This consisted of a fecal pellet from strain of mouse naturally lacking Enterobacteriaceae. We added in either Colibactin-producing E. coli or an isogenic mutant. By monitoring production of that side product from the CLBP reaction, we can see that indeed our inhibitor is effective at inhibiting activation even in this complex setting. Perhaps most importantly, we also showed that inhibitor treatment can prevent the genotoxic effect of Colibactin when they are co-cultured with host cells. So treatment with the CLBP inhibitor prevents cell cycle arrest. It also prevents the activation of interstrand cross-link repair pathways, and we measure that by assessing ubiquitinylation of a protein called FANCD2, which is part of the Fanconi anemia pathway that repairs interstrand crosslinks. So we're now excited to use this compound in animal models to try to gain mechanistic information about Colibactin's role in cancer, particularly the timing of the genotoxic insult, because we can control the timing of small molecule administration. We also think that these efforts might inspire therapeutic strategies for cancer prevention and interception, particularly perhaps in patients who are at high risk for cancer development. But our efforts to manipulate Colibactin production also raised the question of what this molecule is doing to the rest of the microbiome. DNA-damaging natural products are made in many microbial habitats, and they're often assumed to possess antimicrobial activity. But when current grad student Joelle and former postdoc Justin explored this with Colibactin producers, they found that these organisms possessed very limited antibiotic activity in various culture settings. So to determine whether Colibactin could even cause DNA damage in other bacteria, they performed a simple experiment. They co-cultured a Colibactin producer with an isogenic reporter strain of E. coli that was engineered to respond to DNA damage. So this experiment revealed that Colibactin was indeed damaging DNA in neighboring bacteria. And so there could be many possible consequences of this type of DNA damage, but what intrigued us was the possibility that exposure to Colibactin might be inducing prophages, so latent viruses encoded in neighboring bacterial genomes. So indeed, the reporter strain that we used contained the immunity region from lambda-phage, a regulatory element that turns on the expression of phage-lytic genes in response to DNA damage. And so to really test the hypothesis that exposure to Colibactin might result in prophage induction, we co-cultured a Colibactin producer or a mutant with a lambda lysogen and measured the activity of culture supernatants towards a susceptible E. coli strain. And we found that Colibactin production increased phage titers 1,000-fold compared to co-culture with a PKS mutant. And this effect was abolished when the susceptible strain was missing the lambda-phage receptor. So Colibactin can induce prophages in at least a closely related organism. How broad would the effect of this be? So to examine this, we co-cultured Colibactin producers with a variety of both gram-negative and gram-positive lysogens, and we observed induction of prophages in all of these co-cultures, suggesting the effect could be quite general. We showed that prophage induction by Colibactin can have functional consequences by co-culturing the Colibactin-producing strains with a strain of Citrobacter rodentium encoding Shiga toxin on a prophage-associated element. We showed that indeed toxin production increases in the presence of the Colibactin producer compared to a mutant that can't make this natural product. This suggests maybe microbiomes that contain Colibactin producers might result in differing amounts of toxin production. But that idea, of course, raises another important question. Does prophage induction by Colibactin even occur in a more complex microbial community? And so to examine this question, we incubated Colibactin-producing organisms and various individual lysogens in our complex mouse microbiome sample ex vivo. We then enumerated phage particles using a variety of different methods, and again, we observed that in almost all cases, there's an increase in prophage induction when the community contains a Colibactin producer. So we think that these results open up some interesting questions. So we've basically demonstrated that production of a DNA-damaging natural product in a complex community can induce prophages in neighboring bacteria. Obviously, our understanding of this is still in its early stages, but I think this system provides a nice platform for investigating the ecological roles and implications of Colibactin production and its impact on phage behavior in microbiomes. And to me, this raises a very interesting question of whether additional types of DNA-damaging natural products or other types of natural products are mediating interactions between bacteria and phages in unexpected ways. And so to just summarize the Colibactin story, I've told you about how my lab used a chemical understanding of enzymes to initially connect these uncharacterized genes in the gut microbiome to this complex genotoxic natural product, Colibactin. Elucidating Colibactin's biosynthesis and structure allowed us to uncover its ability to alkylate host DNA, helped us to devise strategies to prevent its production, and finally is taking us in some unexpected new directions, providing an opportunity to study how natural products may be mediating bacteriophage interactions and shaping microbial communities. And so I hope that this story has convinced you not only that microbes are amazing chemists, but also that their metabolic capacities are really powerful catalysts for biological discovery. And this work has also served as an inspiration broadly for other work in my lab and is shaping where we're going in the future. So to give you a sense of some of that future work, one question we obviously have is related to the instability of Colibactin and the fact that it couldn't have been discovered using any types of conventional methods. This makes us wonder what other chemically unstable microbial natural products exist. How do we discover them? What might be their biological roles? This challenge has inspired current graduate students, Devin and Katerina, to begin developing chemical probes that can react with otherwise unstable microbial natural products, trapping them and enabling their discovery. So applying this strategy has recently revealed diazo-containing metabolites from an unexpected group of organisms, pathogenic nocardia strains. These diazo compounds are best known currently for their roles as reagents in synthetic organic chemistry. So the fact that human-associated microbes appear to be making them raises really interesting questions. Our work on Colibactins also highlighted the importance of studying metabolites in complex biologically relevant settings. And we also want to begin to discover new microbial metabolites directly in these types of settings. This goal has inspired a current postdoc in the lab, Jared, to develop an iterative metabolomics workflow that we've been applying to study and identify microbial metabolites that are important for bloodstream infections. We think this type of workflow could also be applied to guide future natural product discovery efforts. And then finally, we also hope to apply lessons learned from studying Colibactin and its role in the gut microbiome to discover enzymes and metabolites in another incredibly important and understudied microbial community within the human body, the vaginal microbiome. Current graduate student Dominic and former graduate student Paula have taken on this goal and have recently discovered some vaginal bacterial enzymes involved in nutrient acquisition and microbe-host interactions from both vaginal lactobacilli and anaerobes. And we hope by understanding this type of metabolism, we'll begin to learn how we can manipulate the composition of the vaginal microbiome, which is strongly linked to health outcomes. So now to end, I just want to emphasize that we have a lot left to do as a community to understand microbial functions, including metabolism. As I highlighted at the beginning of the talk, the majority of the genes in the average healthy gut microbiome can't even tentatively be assigned a function. And this is, of course, true for other microbial habitats beyond the gut. So exploring and deciphering this microbial dark matter is absolutely critical if we want to understand how microbes shape our health and our environment. And I hope the presentation today has convinced you that chemistry can play a central role in this effort. And so with that, I would like to just briefly thank the lab. I tried to acknowledge individual trainees who contributed to these projects as I went along, but I'm grateful for the whole group who encompass every discipline from chemistry to biology, who really are incredibly creative and make our lab a wonderful community with which to do science. I'd like to thank our many collaborators, again, who I highlighted as I went along, our sources of funding. And thank you again for the opportunity to be here. It's wonderful to be able to share my science, and I would be excited to answer any questions. Great. And so one quick note about questions. I've been asked to remind people we have microphones in the aisles, and that if you could use them for questions, that would be wonderful. Hopefully they're on.
Okay. Hello. Thank you. It's very. So my question is that it looks like Colibactin being synthesized and then been released to the extracellular. Mm-hmm. So I'm very curious about what would be the mechanisms for Colibactin to pass through the membrane, and also for what mechanism for the PSK positive cells that they don't uptake the Colibactin. Thank you.
All right, so those are wonderful questions. So to answer the second question first, why don't the bacteria uptake producing bacteria uptake Colibactin? So it turns out they actually do, and they have a second resistance protein that appears to destroy the cyclopropane warhead if Colibactin gets inside. So the first question, how is Colibactin entering other cells, is really intriguing, right? You have an ability to damage DNA in mammalian as well as a broad range of other bacteria. And we have a large natural product that would appear to be of a size and have physiochemical properties that would make it difficult to enter even mammalian cells. So we don't know. We're actively investigating this. One hypothesis we have is that there might be some kind of transporter, that Colibactin might be mimicking some kind of substrate that is actively brought into cells, but obviously much more work to do.
Thank you for the great talk. I just wondered, when Colibactin is inducing all these phage, doesn't this then make the strains that produce Colibactin at risk of destruction from the phage that are induced? Have you thought about this?
So that is also a wonderful question, and we have been thinking about this. And so, one hypothesis we have just from kind of examining the genomic contents of the producing organisms is that Colibactin producers are often lysogens themselves, and so maybe they're protected from further phage infection. But another intriguing hypothesis is that perhaps there's a chance the Colibactin producer might benefit from a subsequent lysogenic infection and might acquire genes that might help it in the future from sort of triggering a phage infection. These are hypotheses that we're excited to explore experimentally.
Thank you.
All right. So in your talk, you're talking about it's very easy to degrade, it's very hard to isolate. So I was wondering whether you actually can do some IEDDA, as you illustrate example there. You can add a chemical probe to try to say when it gets degrades, what kind of transporter is talking to, how it talks to other enzyme, maybe different cell localization, stuff like that. Did you ever try that?
Yes, so those are great suggestions. We have thought about, but not yet succeeded in engineering Colibactin itself to have some kind of reactive functional group in it that would allow us to track it. I think that would be very promising, exciting. We and others have tried various strategies to trap functional groups in Colibactin, and it's been surprisingly unsuccessful. So one can isolate, the Watanabe lab in Japan has shown one can isolate very minute quantities from thousands of liters of E. coli that are derivatized and are also decomposed in other ways. But to me, that speaks to perhaps something we don't yet understand about Colibactin transport and perhaps how it might be protected in some way that we don't yet understand.
Thank you so much.
Do you think it might be possible that your inhibitor would have antibiotic properties?
So that is certainly something that we have considered. Does our inhibitor actually have any negative effects towards bacteria or the mammalian host? And what we've done so far was to test for antimicrobial activity across some representative isolates from the major gut bacterial phyla, found no growth inhibition. We also did some activity-based profiling to try to see in a very coarse way if there was evidence for it interacting with any serine hydrolases in either mammalian or E. coli cells, and we were unable to see any other targets. The D-asparagine residue, which is the key structural motif that's recognized, is very unusual, being a D amino acid. Few other peptidases recognize that group. But I would definitely emphasize we haven't completely ruled out the possibility there could be other targets in bacteria that we haven't found.
Hi, that was a great talk. My question is about the enzyme assembly line. And specifically, when you mentioned that Sam was working in this new biological mechanism, I was wondering if you guys are exploring any other microbes to see if it's also working in this way in these other microbes, or if this is specific to this Colibactin.
Oh, thank you for that question. That is a great idea. It was something we tried to do at the beginning, but found very hard because SAM is an enormous amino acid. Compared to the size of the building blocks normally used by these assembly lines, the side chain is big. And so all of the algorithms that have been developed to predict the specificity of what assembly line building blocks are being used sort of could not handle the sequences of this assembly line component. But I think now with AlphaFold, based predictions and the ability to actually predict structures with SAM bound, I think we'll be able to solve that problem, elucidate the bioinformatic signatures of SAM binding and incorporation, and do that type of analysis would be very exciting.
Thank you so much.
Hi there, great talk. Within PKS, do you see different levels of expression of each component within there, either compared to each other or within different environments, whether it's surrounded by other bacteria or human cells?
Mm-hmm. So yes, another really excellent question about regulation of expression of these genes. So that's been investigated by the Allswald lab early on, and then by a number of other groups since. And various factors impact the PKS gene cluster, and different parts of the gene cluster represent different regulatory units, but they all appear to be fairly simultaneously expressed. They are expressed under conditions of nutrient deprivation. So when cultures begin to reach stationary phase, when iron is depleted, certain types of carbon sources will induce stronger expression. But I would say this has not yet been studied in more complex settings. So I think it will be really interesting to see if there are things like diet and other members of the microbiome end up impacting how much Colibactin is produced.
Thank you.
Yeah. Okay, I think then we're out of time. I'd be happy to stick around afterwards for a few more questions. But thank you all again for the opportunity to be here.
Congratulations, first of all, to all the awardees. And I want to thank Drs. Bertuzzi, Colwell, and Balskus for just wonderful presentations that are kicking off what we think is going to be an outstanding meeting for 2024. On behalf of the ASM Program Committee, it is my pleasure as clinical co-chair to close out this session and invite all of you to attend the welcome reception right outside these doors right when we're done. Before we wrap up and enjoy the networking, please give a round of applause to recognize Dr. Carla Satchel as she concludes her 2-year term as Basic Sciences co-chair on the ASM Program Committee. Thank you very much for your leadership. I've really enjoyed working with you these past several years as co-chair and many years prior to that as team members and track leads for the various tracks for Microbe. Now, as I draw close, as her term draws close, please join me welcoming Dr. Pamela Brown as the incoming co-chair for the Basic Sciences. Currently, Dr. Brown is serving as the MVP track leader, and I am excited to continue collaborating with her as she takes on her new role in this program committee. So to end this, I want to thank you all for attending the opening session. We look forward to seeing all of you throughout the course of the meeting. Feel free to come to us anytime if you have any questions, comments, and enjoy the meeting. Thank you.